Good morning, this is Dr. Rajesh Yadav from Department of Biochemistry. Today we will discuss about the acid-base balance, maintenance and diagnostic test. So we have studied about the acid-base definition, acidic and basic nature of the compounds, determination of the pH, numerical base problem, log formula and determination of pH, and we have already done the practice of the numericals. Now today we will study about the definition of the buffer, buffer capacity, mechanism of the buffer, intercellular and extracellular buffer, acid-base balance, acid production inside the body and regulation of the buffer by the kidney and lungs. So it is the introduction of the buffer. So what is a buffer? The solution which resists changes in the pH by addition of acid and base. It is called buffer solution. So buffer solution was given by the Walter Nernst, and for this discovery he was given the Nobel Prize in 1920. Now buffer capacity can be defined as ability of the buffer to resist changes in the pH when acid or base is added. Now why to regulate this buffer solution? So the buffer solution is used to maintain the homeostasis and to regulate the enzymatic function. So now classification of the buffer. So buffer can be classified in the two ways, extracellular buffer and intracellular buffer. Extracellular buffer containing carbonic acid, bicarbonate buffer and protein buffer, while intracellular buffer consists of phosphate buffer and protein buffer. So protein buffer is considered extracellular buffer and intracellular both. So let's discuss the mechanism of the buffer system. So in the body, there are many types of the buffer system present. I would like to explain the buffer system with a small example, like the combination of acetic acid and sodium acetate is act as a buffer system. So what will happen when the strong acid like hydrochloric acid is added? So if we add the hydrochloric acid, so the buffer part, that is sodium acetate, that will combine with the hydrochloric acid and form the weak acid. In this way, the effect of the hydrochloric acid is neutralized because it is converted into the weak acid. In the same way, if the strong base is added, then what will happen? So if the strong base is added, then the part of the buffer, that is acetic acid, it will combine with the sodium hydroxide and form the sodium acetate and water. So in this way, the strong base is converted into the water and sodium acetate weak salt. So in this way, the buffer solution resists to change in the pH of the solution. So another buffer like extracellular buffer. So in the body, in the cells of the human, carbonic acid plus sodium bicarbonate combination act as a buffer solution. So what will happen if hydrochloric acid is added inside the cells? If a small amount of hydrochloric is added, so sodium bicarbonate of the buffer solution will combine with the hydrochloric acid and neutralize its effect, because it converts the hydrochloric acid into the weak carbonic acid. And in the same way, if the sodium hydroxide is added, so the carbonic acid part of the buffer solution bind with the sodium hydroxide and convert the strong base into the weak base like sodium bicarbonate plus water. In this way, the effect of the base will be neutralized. Next, intracellular buffer. So in the intracellular buffer is a phosphate buffer or orthophosphoric acid S3PO4 plus sodium dihydrogen phosphate. So the combination of this orthophosphoric acid and sodium dihydrogen phosphate act as intracellular buffer. So what will happen then? Hydrochloric acid is added. So sodium dihydrogen phosphate combined with the hydrochloric acid and form S3PO4. And in the same way, when strong NaOH will be added, so NaOH combined with the orthophosphoric acid and form sodium dihydrogen phosphate. In this way, the effect of the base will be neutralized. So what will happen when acidity or basicity will be increased? So the neutral pH 7.4, but if pH is less than 7.35, it's called acidosis. When the pH below 7.25, in the central nervous system, depression and coma occurs. When pH above 7.55, hyperactivity, tetany, and when the pH greater than 7.5, it causing the death. Also, when the pH less than 7, it also causes death. Now we can understand how acid produced inside the body. So there are some metabolism through which acid is produced inside the body. So the carbonic acid is produced 60 to 80 milliequivalent per day, because when the carbon dioxide combined with the water and form the carbonic acid inside the cells. And 1 mole of the glucose produces 2 mole of lactic acid. Sulfur protein gives 3 hydrogen sulfuric 3 gram sulfuric acid. Phosphoprotein and nucleoprotein like 3 gram orthophosphoric is present through the orthophosphoric acid produced from the phosphoprotein and nucleoprotein, and hydrochloric acid, which is produced by the oxyntic cells in the stomach. Now we will discuss about the protein buffer system. So buffering capacity of the protein depends upon the pK value of the ionizable side chain. The most effective group is histidine imidazole group. Its pK 6.1. And in albumin 16 histidine is responsible, while in the hemoglobin 38 histidine is responsible for the protein buffer. So protein buffer is depend upon the amino group and carboxyl group present in the protein. So amino group is attached to the amino terminal. Neutral pH. At the neutral pH, amino group is actually NH3 positive rather than NH2. And in acidic medium, it becomes NH3 positive. While in the basic medium, it becomes NH2. In the same way, at neutral pH, the carboxylic ion is present in the form of carboxylate COO negative instead of COOH. In acidic medium, it becomes COOH, and in basic medium, it becomes COO negative. So we can categorize the buffer into the two categories, like chemical buffer system and physiological buffer system. So chemical buffer system is considered as first line of the defense against pH shift, while physiological buffer is considered second line of the defense against pH shift. So chemical buffer including bicarbonate buffer system, phosphate buffer system, and protein buffer system, while physiological buffer considering respiratory mechanism and renal mechanism. So that means kidney and lungs also regulate the buffer pH. So these are considered as second line of the defense. Now we should discuss about the respiratory regulation of the pH. So how the lungs control the pH. The second lines of the defense against change in the pH. The respiratory regulation of pH is achieved by changing in partial pressure of carbon dioxide. The rate of the respiration is controlled by the chemoreceptor, which is present in the respiratory center, which are sensitive to change in the pH of the blood. Respiratory center in medulla cause hyperventilation, which eliminate more amount of the carbon dioxide. In this way, it controls the acidity in the cells. So, if we are running, then the metabolism will be increased and more carbon dioxide will be produced inside the cell in compared to the oxygen. So, in order to remove this carbon dioxide, lungs start hyperventilation. As a result, increased carbon dioxide will go outside of the lungs. And in this way, lungs regulate the pH and act as a buffer system. Now, renal regulation of the pH. So, how the kidney regulate the pH? So, in the tubular cells of the nephron, carbon dioxide and water which combine in the help of enzyme carbonic anhydrase and form carbonic acid H2CO3, which will break down in the hydrogen and bicarbonate ion, and hydrogen ion come into the tubular lumen, and in this way hydrogen will remove through the body and it regulates the acidity. While kidney also control the basicity, in the tubular cells, glutamine is converted into the glutamic acid with the help of enzyme glutaminase, and ammonia will be released. So ammonia is basic in nature, so it will increase the basicity. So tubular cells release the ammonia, and this ammonia will come into the tubule lumen and combine with the hydrogen ion and form ammonium ion. And in this way, ammonia ion will remove through the body. In this way, kidney regulate acid and base balance, and kidney regulate the pH. Now that we can study about the regulation of the water. When the inside the water the insufficient water is present, then decrease of the blood volume. As a result, decreases blood pressure, which increase angiotensin second, which stimulate the thirst center in the hypothalamus. As a result, increase the thirst, and we take the large amount of the water, which decrease the blood osmolarity. The second things, when the deficiency of the water occur, second mechanism, it increase the blood osmolarity. So blood osmolarity is the concentration of the blood. So when the deficiency of water occurs, the concentration of the blood will increase, that is osmolarity will be increased. As a result, dry mouth occur and osmoreceptor will activated in the hypothalamus. The both action combined stimulate the thirst center in the hypothalamus. As a result, it increase the thirst and water will be taken and decrease the blood osmolarity. In this way, the water is balanced inside the body. So this is the reference. Thank you.